Alright. Good morning. We're going to talk about Chapter 3 today in this presentation, and we're just going to talk about cells and their structure and some common features to all cells. We won't be very specific in cell types. We won't differentiate, say, a nerve cell from a bacterial cell or specific features, but we'll just talk about generally what is common between all living organisms. To remind you, I'll be working using the handout that I've given for you. You can find it in D12, or if you're in the Canvas test pilot, you'll find it under the Unit 1, Chapter 3 materials. Okay. So we're going to talk about cells in general. We became somewhat familiar with cells when we just introduced our biology class. So we talked about the different domains of life, the bacteria versus the archaea versus the eukarya. Before we can get into more specifics, we need a little bit of history though. And without getting too detailed in the history, I'd like to at least point out this thing called the cell theory. And to remind you, a theory is a piece of scientific information that's been accepted and concluded by many scientists to be true. Just like all theories, not everybody is going to believe in all theories, so there's certainly going to be an opposing view to this. However, we're going to utilize the cell theory as a platform for this class. It makes the material kind of fit together and cohesive and actually makes sense. And so we have to go back to the mid-1800s. This collective work through Schleiden and Schwann helped form this cell theory. Schleiden was a scientist who was studying plant material, and Schwann was a scientist studying animal material. And collectively, in their independent studies, what they found is that plants and animals had these repeating structures within them called cells. Okay, so what they said is all organisms are comprised of at least a single cell. Not only should something that's living have at least a cell, the cell should actually be the basic repeating subunit for all living organisms. Okay? In an historical perspective, anything considered to be living at this point was just plant and animal matter. Okay, the diversity of the microbes, etc., weren't fully characterized yet or really understood yet. However, certainly these cell theory tenets do apply now to bacteria and archaic organisms. All right, so these were the first two tenets proposed in the cell theory. The cell theory was modified a few years later through the collective work which is brought through with Virchow in 1855, and he came up with this tenet. He didn't create it, but through his work it made this tenet accepted, is that cells come from pre-existing cells. So basically, in order for a cell to survive, it's got to divide and continue its life cycle. Maybe makes sense to you now. However, if you look at it in a historical perspective, this was not known by any means, because a cell hasn't always been understood, but people used to believe that life would just spontaneously arise. So this tried to help dispel that concept of spontaneous generation. Okay, so again, the cell theory collectively tells us if something's living, it's got to have a cell. The cell should be the basic repeating structure of that living thing, and then that cells come from pre-existing cells, or cells divide. Just a quick illustration of some of the work of Virchow, some of his drawings of the different types of cells that he was looking at. If we look at all cells, there are some common features between all of them. And what do we mean as all? Like domain archaea, bacteria, or eukaryotic organisms. Whatever cell type you're looking at, the cells are going to have DNA. The complexity, the volume, etc. will be variable, but there will be nucleic acid, deoxyribonucleic acid, inside of all cells. Okay, and I know you've heard of DNA, and you have a little bit of an idea about it. And what it does is provide genetic information for the cell. In addition, it regulates cell processes. DNA is critical to cell survival. Okay, so DNA will be found in all cells. Secondarily, there's another nucleic acid called RNA, ribonucleic acid, and it helps. Its primary job is to aid in protein synthesis, right, for manufacturing proteins. RNA is involved in this process. And of course, proteins. Okay, proteins, as we studied in the second chapter, are critical for bringing about cell work. And so they're there inside of the cell. We could just list for, I think, 20 days straight what a protein might do for a given cell. Okay, there's so many different types. And certainly ribosomes are critical because they're going to work with DNA, they're going to work with RNA to help manufacture protein. So these are also involved with the production or the synthesis of proteins. And we can't forget, of course, the membrane of a cell. We call it a cell membrane. I'll refer to it as a plasma membrane. But the primary job of the plasma membrane is just to define the boundaries of the cell. But it certainly is very critical for regulating what enters and exits the cell. Okay, so that's common to all cells. Of course, is identified in DNA, but it would apply to all the features, the structure. It's going to be slightly variable between the diverse types of cells that live here on Earth. You should recall from our organization of our life that we have cells will either be prokaryotic in nature or eukaryotic in nature. If you remember, our prokaryotic organisms are domain archaea and our domain bacteria. Conversely, our eukaryotic organisms, they fall collectively in the domain eukarya, and they had the subkingdoms as well. They're kingdoms. We had the kingdom Plantae, kingdom Fungi, kingdom Animalia, and kingdom Protista. What we're going to do is talk about some primary differences between prokaryotic and eukaryotic cells, and then for the rest of this class, we'll kind of focus on eukaryotic organisms mostly. Okay, so if you turn to page 2 of your handout, we're going to compare and contrast some primary differences between a prokaryotic and a eukaryotic cell. The prokaryotic term tells us pro means before and karyo means nucleus. These are the cells that lack a nucleus. Okay, they have DNA, don't misunderstand that. They have DNA, but it's not just compartmentalized in a membrane-bound nucleus, so they just don't have a nucleus. Prokaryotic cells as a whole are much more microscopic when contrasted to a eukaryotic cell, and a prokaryotic cells will never form a multicellular organism. They exist, the whole organism is the cell itself. I have a term here, lacking most organelles. I think that's the phrase that your book uses, and I want to clarify this for you. They lack any membrane-bound organelles. There is no internalized membrane inside of a prokaryotic cell. Okay, we already listed, like, there is no membrane around the DNA. There's no nucleus. There's no mitochondria. There's no chloroplast, etc. However, we cannot forget that ribosomes are in prokaryotic organisms. Ribosomes themselves do not have a membrane around them, but some people categorize a ribosome as an organelle. So, depending on your definition of an organelle, would dictate how you might say what a ribosome is. For our class, we're going to define an organelle with something that has a membrane around it. So therefore, categorically, a ribosome is not an organelle by our standpoint. Okay, but if you think that ribosome is an organelle, by all means, you're just as right as me saying that it's not. Okay. If we contrast eukaryotic cells, eukaryotic, the EU stands for true, and the karyo is talking about the nucleus. So your eukaryotic cells have a nucleus. Okay, so that means is, yes, they have DNA, but there's a membrane compartmentalizing it. And we'll study the nuclear structure coming up in this chapter. As a whole, eukaryotic organisms are much larger, like folds larger than a prokaryotic cell, so they're generally easier to look at under a microscope. Eukaryotic cells or organisms are often multicellular. Okay, so for example, you or the grass in your yard, okay, multicellular organisms. However, not all eukaryotic organisms are multicellular. There are some small aquatic eukaryotic organisms that are just single-celled, and they exist right in their little aquatic environment and are able to survive as a single cell. Eukaryotic organisms do have many membrane-bound organelles, and that's what we're going to study today, their structure and their function. Got a quick video. Hopefully I can get this to work in the recording for you.
Everything that lives is made of cells, even us. Although we can't see them, trillions of cells within our body work together to give us the gift of life. These tiny units of life fascinate biologists like Boyce Rensberger.
The entire human body is made out of cells, or the things the cells have made, such as bone. Skin is made of cells. Your heart is made of cells. The muscles that operate the body so that you can move, these things are all made of cells, including the brain itself, which sends electrical signals around from one cell to the other so that we can think. All these different kinds of cells work fundamentally the same way. When you look inside the cell, you see the same kinds of structures.
Cells come in all sorts of sizes and shapes. They're so small that we need a microscope to see them. Inside of every eukaryotic cell is a microscopic factory that runs off chemical reactions. At the center, the nucleus acts as the cell's brain and controls its activities. It also holds the DNA, the instructions for life. The cell's membrane protects each cell and takes in the raw materials it needs to stay alive. Water, nutrients, and waste move into and out of the cell all the time. These materials may hitch a ride along the cell's transport network, a network made up of microtubules and intracellular membranes. The cell's transport network picks up, transports, and delivers material throughout the cell. The cell breaks down raw materials into the building blocks it needs to grow. This ability to take in vital materials allows the cell to function and perform specific tasks.
The instructions that are in the cells, starting in embryonic development, tell each cell how to change to take on a new function. Also, the cells monitor the activities going on. If there's a wound, the cells can sense that, and they will send out the message to other cells to come and repair the damage. So all of this keeps the whole body on a kind of an even keel.
Our cells work together in extraordinary ways. The heart, for example, is made up of muscle cells with a tendency to twitch. Separately, they twitch to their own rhythm, but together they beat as one. Millions of them work together to give us a heartbeat. These muscle cells survive as long as we do, but throughout the body, cells die and get replaced all the time. Our body produces new cells through mitosis, the process where one cell divides into two. Since we're constantly losing and replacing cells, most cells in our body are actually younger than we are.
Okay, hopefully that video worked in the screen capture. I'll know if you guys tell me if it does or does not. But anyway, that was a nice little video introduction into just basic features of the cell. To remind you, we are going to talk predominantly about eukaryotic cells this semester. And we're going to compare and contrast a typical animal cell to a typical plant cell, because those are two types of organisms that you as a person in society could easily tell the difference from, right? You could tell the difference between a plant out in the environment in an animal. So those are common things that you'll run into, so we'll kind of focus on those types of cells. And generally, beyond like photosynthesis, we'll kind of talk about what happens inside of the human body, because again, that's really something that you could understand better. Here's an example of a typical textbook looking animal cell. This is super generic. I mean, it's generic. I mean, just the word is described as this cell. Animal tissues, right, are very unique. I keep making reference to, for example, nerve cells, right? Nerve cells definitely don't look like this. However, they would have all of these features within them, as well as, say, your skeletal muscles. Your muscles look profoundly different from this, but they would still have these same structures within them. So we're just looking at a generic looking cell. What would each of these components do and for a given cell? So we're going to go through and look at these different organelles and structures inside of the cell. We'll end up contrasting those to a plant cell and find some features specialized in plant tissues as well. The first thing that I want to talk about is the cell's plasma membrane. That's the boundary, or you can call it the cell membrane, and that's the boundary for this cell. And as well as membrane will surround each of our little organelles inside of a given cell. So if you look at the top left image, it's actually a little better visual detail of the features that you would find inside of a membrane, and you contrast that to the image on the lower right hand side. If you look at the membrane there, it's really kind of plain and boring, right? But if we look at it in the top left image, we can actually see that it's a complex structure. I would like for you to also realize that the membrane, when we talk about it, the cell, boundaries, right? Similar features when you're looking at it in a mitochondria or a lysosome or a peroxisome, etc. Okay? If we look at the plasma membrane, what it does, as I identified earlier, it's just, besides just providing a boundary for the cell, it regulates what enters and exits a cell. Okay? Because a cell has to, as you identified earlier in this semester, has to be responsive to its environment. And therefore, particles need to enter, right? For example, in your body, if you eat food, those nutrients need to be broken down and taken into your individual cells. But just like at your house, you bring in food, right? There's going to be waste products. You should take that waste product out of the house. So same thing inside of the cell. When food comes into the house, into the cell, then waste products should also be shipped out of the cell. Okay, so certainly regulating what enters and exits the cell, as well as the cell membrane allows for cells to interact with other cells. You may remember from the video that I just showed how a man talked about if there's injury, the cells can recognize and kind of bring in help. It's due to the complex set of molecules incorporated in the cell membrane that allows for that kind of cell talking or cell interaction. And boundaries, already identified that. If we look at the generic structure of the plasma membrane, you're going to find a bunch of molecules in there, and collectively you'll find phospholipids, different types of proteins, other classes of lipids, these things called glycoproteins. We've not used that word yet, but basically it's a protein with a sugar attached to it, as well as glycolipids, which you can take away as a sugar and a lipid molecule attached. I don't believe we use the word phospholipid either in Chapter 2. A phospholipid is very similar to a triglyceride, except for instead of having 3 fatty acids, a phospholipid actually just has 2 fatty acid tails. And again, the degree of saturation or unsaturation is variable. But collectively, right, we have a mixture of these molecules making up the membrane. The bulk of the membrane structure is arranged in what's called a bilayer. And as the name implies, bi, right, 2. There's 2 layers of lipids arranged. And we have what's called a hydrophobic and a hydrophilic area when we have this bilayer arranged. I know that we use these words when we talked in Chapter 2, Features of Water. And just to recap for you, hydro, the prefix means water, phobic means fearing, and philic means loving, right? So what that tells us is the lipid bilayer has an area that is water soluble and an area that is water insoluble, okay, or water fearing. probably better to understand if we look at the image shown now, or you can see on page 2 of your Chapter 3 handout. The generic looking phospholipid bilayer. There are no additional molecules, there's no proteins, there's no glycolipids, none of those other components are shown here. But we just want to understand the phospholipid features. And here we can certainly see the 2 fatty acid tails, okay, looking up, 1, 2, of each of, let me get these colors right, of each of the little fatty acid tails. So 1, 2, right? That with a little head group would be a single phospholipid. Okay, so on the top we have one layer, and on the bottom we have a second layer. Just to help you put it in perspective, each little blue sphere with the gray tails is a phospholipid, and then therefore we have 1, 2, 3, 4, 6, 7, 8, 11 phospholipids adjacent in the top layer, and as well 11 on the bottom layer. Let's look at a little better detail of the phospholipid itself, though. You should recall that fatty acids are hydrophobic, and so you can see the fatty acid tails, when arranged in the bilayer, actually orient towards themselves. And so the greatest thickness of the lipid bilayer is actually the lipid part. Okay? And this is how we create a water boundary, if you will, for the cell. If you remember, the fatty acid tails are hydrophobic, so basically water-soluble particles would have difficulty traveling right from one location to another through this fatty acid layer. Okay. However, there's water around the cell and water certainly within the cell. Within the cell, I have labeled down here ICF, which is just basically, called the intracellular fluid, okay, the fluid inside of the cell. Okay. Let me fix this for you, sorry. Okay, the intracellular fluid, okay, the fluid inside of the cell. Okay. Conversely, there's water outside of the cell called the extracellular fluid. And if you remember, the phosphate containing group of the phospholipid, or the triglyceride, is the example we gave in Chapter 2, is actually charged, okay? And so that it's going to be capable of forming hydrogen bonds with water molecules in either the extracellular fluid or the intracellular fluid. And so what we can say is a single phospholipid is both polar and nonpolar. The nonpolar component would be where the fatty acid tails are, and the polar component is where the heads are. Okay, so now we have a nice little fatty layer that protects, separates the outside from the inside of the cell. We also identify that there are proteins associated with the plasma membrane. In the images below, the proteins are the purple globs. Okay, if we look at, for example, here where if you can see the check mark, labeled number 1, that's a protein. Right, number 2, that's a protein. Number 3. Okay. Even these things in the inside of the cell, the little purple things, those are proteins. Okay. You'll notice that they're not all built the same because they would not have the same function. And you'll notice that some of the proteins have carbohydrate chains branching off of them. That's what we would call a glycoprotein. And some of them form channels. Some of them allow for cell-to-cell interaction. Okay, so we'll get into some more specific roles. But just generically speaking, if you see a membrane protein, it's probably going to transport something across the cell membrane. Like, for example, if you look at the little protein I labeled as number 3, I'll try to highlight. Okay, something polar, charged, normally would have difficulty diffusing through the membrane. However, if we create a little channel, something polar could move in or maybe out of the cell, and we'll talk about what drives that. Okay, so we might be able to transport substances across the membrane. There's also enzymes sometimes associated with the cell membrane. For example, looking on the right-hand side, I'll just draw a little green blob over. They help speed up specific types of chemical reactions. They might aid in cell-to-cell recognition, like we gave an example earlier. And usually it's these glycoproteins, like we have highlighted here as I labeled number 1. You can see the little sugar group projecting out. There would be sugars of another